Imagine a world where the tiniest organisms on Earth, bacteria, something we usually fear and fight, become our most powerful allies against one of humanity's greatest enemies, cancer. What if the solution to a disease that claims millions of lives each year could be hidden in the microscopic world within and around us? Today, we're going to explore a groundbreaking approach where certain bacteria don't just coexist with us, they can actually help destroy cancer cells from within, awakening our own immune system to fight back. This is not science fiction. It's cutting-edge research that is changing the way we think about medicine, the body, and the fight against cancer. You'll discover how these microscopic allies navigate deep into tumors, trigger self-destruction in cancer cells, and rally the body's natural defenses in ways conventional therapies often cannot. By the end of this discussion, you may see bacteria not as something to fear, but as a beacon of hope in modern cancer treatment. Stay with me as we uncover this fascinating journey into a microscopic battlefield where science, nature, and hope converge, showing that sometimes the smallest things can make the biggest difference. Certain bacteria have the remarkable ability to stimulate the immune system in ways that can help the body recognize and attack cancer cells. Normally, cancer develops partly because tumor cells evade immune detection. They disguise themselves or suppress immune responses, allowing them to grow unchecked. Some bacteria, however, naturally activate strong immune reactions when they enter the body. This immune activation can disrupt the protective environment that tumors create around themselves. When specific bacterial strains are introduced in a controlled and weakened form, they can act as biological alarms. The immune system detects bacterial components such as proteins and cell wall molecules and responds by sending immune cells, like macrophages, dendritic cells, and T cells, to the area. If bacteria are present inside or near a tumor, this immune response can expose cancer cells that were previously hidden. As immune cells become activated, they release signaling molecules called cytokines, which further enhance the anti-tumor attack. In some research settings, bacteria have been engineered to increase this immune-stimulating effect while reducing harmful side effects. By boosting immune recognition and reducing tumor-induced suppression, bacterial therapy may transform a cold tumor, one that avoids immune detection, into a hot tumor that the immune system actively targets and destroys. Some bacterial strains are being studied for their unique ability to specifically target tumors within the body. Unlike many conventional treatments that circulate broadly and affect both healthy and cancerous tissues, certain bacteria naturally migrate toward tumor environments. Tumors often contain regions with low oxygen levels, abnormal blood vessels, and areas of tissue breakdown. These conditions create a microenvironment that is difficult for many therapies to penetrate, but can be attractive for particular types of bacteria, especially anaerobic or facultative anaerobic strains. Once inside the tumor, these bacteria can multiply selectively because healthy tissues typically have stronger immune surveillance and oxygen-rich conditions that limit bacterial growth. This selective colonization allows researchers to use bacteria as delivery vehicles. Scientists are exploring ways to engineer these strains so they can release anti-cancer toxins, immune-stimulating molecules, or enzymes directly within the tumor mass. This localized action may reduce damage to surrounding healthy cells while increasing therapeutic precision. In laboratory and early-stage animal studies, tumor-targeting bacteria have shown the ability to shrink tumors by disrupting cancer cell function and enhancing immune system engagement. Researchers carefully modify these strains to weaken their disease-causing potential while preserving their tumor-seeking properties, aiming to balance effectiveness with safety. Tumors often develop regions with very low oxygen levels, a condition known as hypoxia. As cancer cells grow rapidly, they outpace their blood supply, leading to poorly organized and inefficient blood vessels within the tumor. This creates areas deep inside the tumor where oxygen and nutrients are scarce. Many traditional cancer treatments, such as radiation therapy and certain chemotherapies, depend on oxygen or effective blood circulation to reach and destroy cancer cells. As a result, these hypoxic zones can become safe havens where cancer cells survive treatment and continue to grow or spread. Certain types of bacteria, particularly anaerobic bacteria, naturally thrive in low oxygen environments. Unlike normal healthy tissues that contain abundant oxygen and active immune defenses, the hypoxic core of tumors provides a more suitable setting for these bacteria to survive and multiply. When introduced in a controlled and weakened form, these bacteria can penetrate deep into the tumor mass, including regions that standard therapies struggle to access. Once established within these oxygen-poor areas, the bacteria can directly damage cancer cells or stimulate an immune response within the tumor. By targeting the very zones that resist conventional treatments, bacterial approaches may help overcome one of the major biological barriers in cancer therapy. Research in immunotherapy is exploring how modified bacteria can be used as active tools in cancer treatment. Immunotherapy focuses on strengthening or guiding the body's immune system to recognize and eliminate cancer cells more effectively. Some bacteria naturally provoke strong immune reactions, and scientists are studying how to harness this property in a controlled and therapeutic way. By genetically engineering bacteria, researchers can reduce their harmful characteristics while enhancing their ability to stimulate targeted immune responses against tumors. These modified bacteria can be designed to enter tumor tissue and produce specific molecules, such as cytokines or tumor antigens, that alert immune cells to the presence of cancer. They may also be engineered to deliver immune checkpoint inhibitors directly within the tumor environment, helping immune cells overcome suppression signals that normally prevent them from attacking cancer. In addition, bacteria can act as microscopic factories, releasing therapeutic proteins precisely where they are needed. In preclinical studies, such approaches have shown the ability to transform immune-resistant tumors into ones that attract immune cell infiltration. Careful testing focuses on balancing immune activation with safety, ensuring that the immune response remains controlled rather than causing excessive inflammation or systemic infection. Laboratory studies have shown significant cancer cell reduction when specific bacteria are introduced under carefully controlled experimental conditions. These studies are typically conducted in cell cultures or animal models, where researchers can closely monitor how cancer cells respond to bacterial exposure. In cell culture experiments, cancer cells are grown in dishes and then treated with selected bacterial strains or bacterial byproducts. Scientists observe whether the bacteria directly damage the cancer cells, interfere with their growth signals, or trigger programmed cell death mechanisms. In animal studies, weakened or engineered bacteria may be injected into tumors to evaluate how they behave inside a living organism. Researchers measure tumor size over time, assess immune system activation, and analyze tissue samples to determine how effectively the bacteria reduce tumor mass. In some cases, dramatic reductions in cancer cell populations have been recorded, especially in tumors with low oxygen regions that favor bacterial growth. These laboratory findings are important because they demonstrate proof of concept. They show that under specific and controlled conditions, bacteria can significantly disrupt tumor growth. However, laboratory environments differ from the complexity of the human body, so further research is required to confirm safety, dosing, and consistent effectiveness before clinical application. The phrase 99% of cancer cells often appears in headlines or discussions to emphasize dramatic laboratory findings, but it usually refers to results obtained in controlled experimental settings rather than confirmed outcomes in human patients. In many preclinical studies, researchers test bacterial therapies on isolated cancer cells in petri dishes or in animal models such as mice. Under these tightly regulated conditions, scientists can control variables like dosage, timing, and tumor type, sometimes observing very high percentages of cancer cell destruction. However, the human body is far more complex than a laboratory model. Tumors vary widely between individuals in size, genetic makeup, location, and interaction with the immune system. A treatment that eliminates 99% of cancer cells in a dish may not perform the same way in a living human due to factors such as immune variability, metabolism, and potential side effects. Additionally, eliminating 99% of cancer cells does not always mean a complete cure, as even a small number of remaining cells can regrow. Therefore, such percentages are best understood as indicators of potential effectiveness during early research stages, rather than guaranteed clinical results for patients undergoing treatment. The immune system plays a central role in bacterial-based cancer therapies because it is ultimately the body's own defense network that carries out the destruction of abnormal cells. Cancer often progresses by evading immune detection, creating a suppressive environment that weakens immune responses. Tumors can release signals that deactivate T cells, attract regulatory immune cells that dampen attack, or form physical barriers that prevent immune infiltration. Bacterial approaches aim to disrupt this protective shield and reawaken immune activity within the tumor microenvironment. When weakened or engineered bacteria enter tumor tissue, the immune system recognizes bacterial components as foreign. This recognition activates innate immune cells such as macrophages and dendritic cells, which begin producing inflammatory signals. These signals recruit and activate adaptive immune cells, particularly cytotoxic T cells that are capable of identifying and killing cancer cells. As inflammation increases within the tumor, previously hidden cancer antigens become more visible to immune surveillance. By converting an immune-silent tumor into an inflamed and immune-active site, bacterial therapies can enhance the body's natural capacity to attack cancer. The effectiveness of this strategy depends on carefully balancing immune activation so that it is strong enough to target tumor cells without causing excessive systemic inflammation. Bacteria may trigger apoptosis, or programmed cell death in tumor cells, through several biological mechanisms. Apoptosis is a natural and highly regulated process that allows the body to eliminate damaged or abnormal cells without causing widespread inflammation. Cancer cells often develop the ability to resist apoptosis, which allows them to survive longer than normal cells and continue dividing uncontrollably. Certain bacterial strains or their byproducts can interfere with this resistance and reactivate internal cell death pathways. When bacteria colonize tumor tissue, they may release toxins, enzymes, or metabolic byproducts that stress cancer cells. This stress can damage cellular structures, disrupt mitochondrial function, or activate signaling cascades that initiate apoptosis. In addition, bacterial infection within the tumor can stimulate immune cells to release cytokines such as tumor necrosis factor and interferons, which further promote apoptotic signaling in nearby cancer cells. Some engineered bacteria are specifically designed to deliver therapeutic molecules that directly activate apoptosis-related genes inside tumor cells. By turning back on the cell's built-in self-destruction program, these approaches aim to reduce tumor mass in a controlled manner. The ability to selectively induce apoptosis in cancer cells while minimizing harm to healthy tissue remains a key focus of ongoing research. Clinical trials are ongoing to evaluate the safety and effectiveness of bacterial-based cancer therapies in human patients. Before any treatment becomes widely available, it must pass through a structured series of trial phases designed to protect participants and ensure reliable results. Phase 1 trials primarily focus on safety, determining the appropriate dosage, and identifying potential side effects. Because bacteria are living organisms, researchers pay close attention to how they behave inside the human body, including how long they persist, how they are cleared, and whether they cause unintended infections or inflammation. Phase 2. Trials begin to assess how well the therapy works against specific types of cancer while continuing to monitor safety. Researchers measure tumor response, immune activation, and overall patient health. If results are promising, Phase 3 trials compare the new therapy against current standard treatments in larger groups of patients to confirm effectiveness and detect less common side effects. Throughout this process, regulatory agencies review data carefully before granting approval. Clinical trials also help scientists refine bacterial strains, improve delivery methods, and adjust treatment combinations. This step-by-step evaluation ensures that potential benefits outweigh risks before broader medical use is considered. This approach is fundamentally different from chemotherapy and radiation because it uses living organisms rather than chemical or physical agents to target cancer. Chemotherapy works by attacking rapidly dividing cells throughout the body, which often affects healthy tissues such as hair follicles, the digestive tract, and bone marrow, leading to significant side effects. Radiation therapy, while more localized, uses high-energy waves to damage DNA in cancer cells, but it can also harm harm nearby healthy tissue and often requires multiple sessions. Bacterial-based therapies, in contrast, aim to selectively target tumor cells or the tumor environment itself, potentially reducing collateral damage to normal cells. The mechanism relies on the natural behavior of bacteria to thrive in specific tumor microenvironments and to activate the immune system. Instead of broadly attacking all rapidly dividing cells, bacteria can localize to areas where oxygen is low or where immune surveillance is weak, such as deep within a tumor. Once there, they can directly damage cancer cells, release therapeutic molecules, or stimulate immune responses to recognize and eliminate the tumor. This targeted, biologically driven approach represents a new paradigm, combining precision with the ability to leverage the body's own defenses, potentially resulting in fewer systemic side effects compared with conventional cancer therapies. Safety is a critical concern in bacterial-based cancer therapies because these treatments involve introducing living organisms into the human body. Even bacteria that are normally harmless can cause infections or trigger excessive immune responses if not carefully controlled. To address this, researchers use weakened or genetically engineered strains that retain tumor-targeting and immune-stimulating properties while minimizing their ability to cause disease. These modifications can include removing genes responsible for toxicity, adding safety switches that allow the bacteria to be eliminated if needed, or engineering them to die after a certain period. Monitoring patient responses is also an essential part of maintaining safety. Scientists track immune reactions, inflammation, and the distribution of bacteria within the body to ensure that the therapy is localized to the tumor and not affecting healthy tissues. Dosing is carefully calculated, often starting with very small amounts and gradually increasing under controlled clinical trial conditions. Additionally, combination strategies may be employed, such as using antibiotics to control bacterial populations if unexpected side effects occur. The goal is to balance therapeutic effectiveness with safety, ensuring that the bacteria can attack the tumor without posing significant risks to the patient. This careful design and monitoring are key to making bacterial therapy a viable and clinically acceptable approach to cancer treatment. While bacterial cancer therapy shows great promise, it is still an area under active scientific investigation and not a guaranteed cure. The research is largely in experimental or early clinical stages, meaning that most findings come from laboratory studies or small-scale trials rather than widespread use in humans. Scientists are exploring which bacterial strains are most effective, how they interact with the immune system, and how to deliver them safely to tumors. This careful, step-by-step process is necessary because human biology is complex, and results observed in controlled lab conditions may not always translate directly to patients. Moreover, tumors vary widely in size, type, location, and genetic makeup, which affects how they respond to bacterial therapy. Some patients may experience strong anti-tumor effects, while others may show minimal response. Researchers are also investigating potential side effects, optimal dosing, and the best ways to combine bacterial therapy with other treatments like immunotherapy, chemotherapy, or radiation. Regulatory approvals require extensive testing to ensure that benefits outweigh risks, which is why bacterial therapies are not yet standard treatment options. Despite the excitement, it is important to maintain realistic expectations. The field represents a shift toward innovative, biologically based approaches, but ongoing studies are needed to confirm long-term safety, effectiveness, and reproducibility before these therapies can become widely available for cancer patients. In conclusion, the use of bacteria in cancer therapy represents a groundbreaking and promising frontier in medicine. By harnessing the unique ability of certain bacterial strains to target tumors, stimulate the immune system, and even trigger cancer cells to self-destruct, scientists are opening new pathways for treatments that are more precise and potentially less harmful than conventional methods. While this approach is still under research and not yet a guaranteed cure, the ongoing studies and clinical trials demonstrate immense potential to transform the way we think about fighting cancer. It highlights the incredible possibilities that arise when we look to nature and innovative science for solutions. Good luck to everyone on your journey toward knowledge, discovery, and health. Thank you for giving your attention and taking the time to explore this exciting field of science with me today.